Yes. So go back. So I should be recording now. So I am recording now.
Okay.
Let me go back. And so you're going to take your template. You're going to save the template as a copy. So file, make a copy. And then you're going to put that. So I put it into Nick's Bio 240. Okay. You're then going on your saved document. You're going to click on the share and make sure that you share it with me. So do share. And then what you can do is, sorry, Karamoko, thank you for reminding me. So share it with Nick Cap, or you can cap at SMCCD. You don't need to notify me on this because I don't need to get 60 notifications that you're doing that. Click on share. Again, realize that when you do this, if you go get the link, change the link. And so this says anyone on the internet with this link can view. I need to be able to add it to documents. So you need to go, instead of a viewer, you need to make me an editor. Commenter may work, but I want to be able to kind of edit your document. So you want to send that to me. And so in this one here, it's saying anyone with the link can do it. You could also say restricted if you're really worried about security, which I don't think you necessarily should be, because you're not really sharing this with other people. But I would say anyone with that link. In that way, both myself and Professor Yang Yuan can open this particular document. Okay. I read through the introduction to this, and then what I want you to do is to record your at-home microbiology workstation. Again, what you're going to do is you're going to have a table here. You're going to have 3 columns in your table. I will give you a task in the first column. In the second column, you're going to describe in your words what you're doing, and then I want kind of a labeled picture of what you're doing. And then the next one, activity number 2, this is basically looking what microbes are in your area. And so you'll take your plate, just open up your plate and go ahead and put it down on your bench top. And what you're going to do is just let it sit for an hour and see what microorganisms fall into there. Again, when we do this in the laboratory, it's really clean and it's kind of hard to see. If you want, you can take things like your cell phone. It won't all fit on there, but it'd say, just, what bacteria on my cell phone? So I dabbed it on there on your cell phone, or you can put your finger on there and you can kind of see what's there. You get 3 plates to do this with. I gave you 5 plates. If one of the plates is contaminated, then you don't necessarily have to do that. If you want to use all 5 plates, you can, but I'd wait for a second. Do that 3 plates first. What I want you to do is to take these plates, invert them, maybe put them in a sandwich bag, put them in a dark, warm place. If you have a 37 degree Celsius incubator, that'd be great. But if most of us don't, just put it in, just put it away. It's warm. Don't put it in sunlight because UV light will affect these. Just put it in the dark, and we're going to let them grow, and we're going to see what's on there. And you're going to record what's on there. So after 3 days, what do your plates look like? Explain what you see. Make sure your pictures are labeled. And we'll do an analysis on this a little bit later. What I want you to do right now is just take a look at those guys. Any questions on that? So that should take about 5 minutes in all to kind of do. The next thing I want to do is look at the effectiveness of hand washing. And so as an introduction, the largest organ on your body is the human skin. There's lots of bacteria there. It provides a physical barrier of protection from pathogens. In addition, its surface is covered with bacteria. There are 2 main types of bacteria that can be found on the surface of your skin. What we call transient bacteria that are present for days or weeks. You can pick them up daily, such as when you shake hands or open doors. These microorganisms have the potential to cause disease, such as Salmonella or Shigella species. Deeper into the top layer of skin are a different set of microbes called resident bacteria. These bacteria are part of your normal flora of our skin and are present for months or longer, sometimes years. They do not normally cause disease, but damage to the outer skin layer may change that. If they get inside, then sometimes they can cause disease. And we'll talk about that a little bit later in class. These resident bacteria play a role in protecting us from pathogens. For example, Staphylococcus epididymis produces bacteriocins, bacterial antibiotics that kill potentially disease-causing bacteria like Staphylococcus aureus. I want you to work individually. And what we're going to do is I do want you to know what cleaning and disinfection are. So this talks about it. So what it's talking about is I gave you a little substance here called glow germ, right? And what I want you to do is to take some of your glow germ. Okay, these were packed aseptically. You could go in there and take about a quarter size of glow germ and kind of put it on your hands, okay? And, while it's wet, well, I touched my thing. Oh, I touched my computer. Oh, I'm touching the tabletop. Okay. So what I want you to do is kind of wipe it in your hand, and you can let it dry for a little bit. It's a moisturizer. I gave you a whole bunch of this, so I have a whole bunch left, but there's a couple things that we can do. So the idea behind this is that stuff can go everywhere. So what I want you to do on your task is contaminate your hands with a quarter-size tab, not bab, of glow germ. Make sure that the lotion dries completely. It's still going to feel a little bit oily. Take a picture to demonstrate the contamination. So I'm going to turn off the lights here. Okay. I turned off the lights. And then you got a little key fob. This is yours to keep. Everything in your kit is yours to keep. We don't want them back, sorry, in times of COVID. So what I want you to do, if you do this, it's kind of hard to see, but you see how it lights up? Kind of hard to see on the screen. So you're going to take your screen. I'm gonna try and shut the door and see if I can do it a little bit better. Okay. You're seeing my screen, but basically what's happening is my hands are lighting up green. And actually, if we go through and look at this, you can also see where I touched my cell phone. See the little green stuff floating up on the cell phone? And then also on the tabletop, you could also see the tabletop kind of lighting up. So this glow germ stuff is safe. It washes off with water and soap. And it's not a microorganism. And what we want to do is kind of demonstrate how microorganisms can kind of get everywhere. And so what I want you to do is to contaminate your hands and then try and get a picture of your hands. And I'll put a demonstration of an example of a picture in there. Let's see if I do insert image, photos. I took some photos last night. And so here's a picture, if you can see it, pop it in there. Okay, so there's my hand.
I can't see it.
You can't see it?
No.
You still have your other stuff up. Oh, okay. There you go.
Oh.
Okay. Do you see in the document?
Yeah. Can you show me how you inserted that picture again, please?
So what I did is, so I have it hooked up so that when I take a picture on my phone, it goes to my cloud site and it saves it there. And so what I did is wherever I want to put it. So let's say I want to put it in this box. I go insert image. And for me, because my photos are all shared, and so here's an example of me contaminating something on a desktop. Okay.
Okay.
So there's this, and then I can change the size of that if I want. And I could crop it, so there's some tools here. We can crop it and do some of the other things on that. If you don't have that access, what you can do is a picture that's on your computer. If I go, let me do this, go insert image, upload from the computer. Here's one I did the other day. So a friend of mine who was volunteering from the Moderna study took a selfie when she was getting vaccinated against COVID. And Okay. So I changed the side. Anyone else? And so I could, if I want to edit that or if I want to do something, so I can do things like insert. There's other things that you can do. So you can label a picture and stuff like that and do a number of different things in there. So let me delete that out. Okay. Any questions on that? So what I want you to do is task to do, and I'll leave those in here because this isn't mine. So that's not in, it's not in the template. So that shouldn't affect you guys. And so what I want you guys to do is for the second one, safety. Take a picture and describe the typical work. So no, this is that workstation. There's safety plans. So you guys did this already. So here's the microbes are everywhere. We're looking for microbes. So you're going to set up I want you to set up an experiment. Sorry. We're on effectiveness of hand washing. And so what I want you to do is contaminate your hands. And so I did this again. So I'll do insert, image. Mine is on my photo Google Doc. And I can do something like this. I put the photo in there. I'm going to resize it. So this is just a picture of my hand that's contaminated. What I want you to do then after that is, so take a picture of your hand. What I want you to do is to rinse your hands and see what happens, right? So when you rinse your hands, I want you to take a picture of it afterwards. Well, what do you think is going to happen? Stephanie, what do you think is going to happen when you rinse your hands?
I think maybe you won't see as much as of the glow germ as you did before.
Yeah, you may not see as much, but probably you're still going to see the glow germ. Yeah. What I want you to do is to soap up and wash your hands. And I should have on the top here, here's a video on how to properly wash your hands. And so what you should do is properly wash your hands. And then, because also, you don't want this glow germ stuff to be on your hands, even though you can't see it. So go ahead and wash that out and then kind of tell me what happens. And then what I want you to do on this last one is compare and contrast the difference in removing contamination on your hands between rinsing and washing soap. So you can put those pictures side by side. For activity number 4, I'd like you to design your own experiment. With the leftover glow germ, that's a bunch, and your UV light, design an experiment that allow you to follow contamination on your workstation. So here's maybe something fun. Trying to have somebody else maybe look at that glow germ, maybe have somebody else put the glow germ on, or maybe you go in your workstation, put the glow germ on, and kind of touch around your workstation and see if somebody else can tell where your stuff is. So here what I want you to do is to set up a hypothesis, set up your results of that, and then give me a description and your pictures. And so, again, I'm in the laboratory at school, and I'm in the laboratory at school. Here's the desktop. And you could kind of see where my palm prints were on the desktop. And again, these microorganisms are no different than the glow germ. Okay, if you have microorganisms, and you're going to have lots of microorganisms on your hand, they, whenever you touch something, those microorganisms can come off and they can move all over the place there. And so what I want you to do is to, design a little experiment. You can use the rest of the glow germ. This is non-toxic. If you don't want to use it, you can go ahead and throw it out and stuff like that. And the only thing it does is it shines up with the UV light. So, even if you get it on something in normal light, you really shouldn't be able to see it. But go ahead and wipe it off. Let me go, I'm going to share my screen again. For the final issue, what I want you to do is I ask some questions, 5 questions. What's the significance of the difference between rinsing and washing with soap? Okay, what's the significance of using soap? Should we use soap? My wife always asks my kids, did you wash your hands? And they all go, yeah. Did you use soap? And they're like, oh, and they have to go back into the bathroom because they wash their hands without soap. Is there a difference? Number two, what did you think the mode of action is for soap? Why does it work? Again, remember here, glow germ's not alive. So it's the soap isn't necessarily killing something. What is it doing? Number three, why is it important that agar cannot be metabolized by many microorganisms? Gelatin was first used to solidify media, but it didn't work. Can you let me know why? And gelatin is just, that gelatin agar that we like to eat. Number four, based on your results, how germ-free do you think your area is? Now we're going to make these areas sterile, but why do you, now we're not going to make these areas sterile, but why do you think your area is good enough to work on? And number five, how do you think you can make your workstation more free of germs? Try and be specific. Look around. Are there any other environmental factors that are involved? What can you do to make it more germ-free? And again, that's kind of one of those things that we tend to think a lot more about now with COVID, right? And so there's these different ways. How do we sanitize our areas so that we don't have to worry about them as much? Okay. We got about a half hour. Sorry, it took a little bit longer. We got about 40 minutes left for you to kind of do this activity. What I'm going to do is once you're done with this activity, there's it's a on the activity, just like a quiz. Crap, I'm going to have to share my screen again. So I'm going to share my screen. If you go under assignments, this will be one of the assignments that you have. Or actually, no, if I go under home, if I go under lab, it is under assignments, go under lab. So number 5, module 5, lab materials. If we click on this, we hand it in. If you click on this, starting it in your home here, if we click on it, all right, so I see something a little bit different than you. But if you click on it, starting in your home, you should be able to turn in that document. So basically, you're turning into me a URL, and I would like you to complete this assignment by, I think I said the 20th. So if you do the assignment today, so you're going to do the assignment, and you're going to wait 3 days before you record your final results. So I'm giving you roughly about a little less than a week to do the assignment. Again, I will accept it, kind of stuff happens up until September 23rd. And so this is covering a couple laboratory periods. So you guys have a little bit of time this laboratory period. And then for you guys, the 24th, we're going to be coming in and we're going to be doing the gram stain experiment. So we're a little bit behind on that. Okay, I'm going to stop share. All right, let me know how it goes. I'm going to be here. Try doing your stuff. If you have something else that you absolutely have to do that. Make sure that you get some time in the next week to do your lab and realize that it's going to take 3 days for your results, for your bacteria to grow. So make sure that you do it you start doing it by at least the 20th so that you can get the growth and record the growth by the 23rd. That being said, I am going to kind of let you go and do whatever you want to do. If you have questions, if you're setting up your lab, or if you want to show me what you're doing in your lab, I would love to see that now. Again, I have no problem if you have people in your household, if they will behave a little bit better, if they do the experiment with you, I am okay doing that. This is all food grade kind of stuff, but just kind of realize that you are the one who are responsible for it. So I know some of you may have little siblings or little kids kind of hanging around, and normally we wouldn't do it, but again, this is time of COVID. If we'll settle them down a little bit by helping you out, then by all means. And even this one here, we're asking you to do an experiment where you're going to need 2 people to work on it. So it's always much more fun to work with somebody in the laboratory. All right, I'm going to turn on my lights. I'm going to be walking around the lab. I'm going to hear if I'm not on screen, you could always say, hey, Nick, and then I should come back. So let me know. Everyone, did you sign in? So you needed to go in and chat and sign in so that I can capture you. I did Zoom this, so I will put it up.
Dr. Cap.
Isabella, hi.
Hi, do you know where we would be able to get a UV light, or is like Amazon probably like the best option?
No, you should have picked one of these
Oh,
up
sorry.
when you came up. So
Okay,
this is
you're right.
yeah, this is the UV light here. You should got one. So I on these things here, most of it you should be able to get from your kitchen. I don't want you to pay, what we really don't want you to pay for that kind of
Okay, I see it. Sorry about that.
stuff. Yeah. So yeah, you should have gotten this in your kit, and I should
Okay.
have made that
Thank
aware.
you.
Okay. Anyone else? Did anyone get any contamination on their plates? What do your plates look like? I mean, you could still use that plates. If your plates have some fuzzy stuff growing on it, you could still use them. Oh, I should have done it. You should be wearing your safety materials. Celia, you had a question or comment?
I just wanted to show you a picture. I don't know if you remember, you gave me an extra set.
Yeah. Send me, if you could, well, if you want to send me a copy of that, I'd appreciate it.
Okay.
I won't publish it because you didn't give me the rights for it, but that's cool.
You can. What's your email? Tell me again.
cap@smccd.edu.
Okay. Thanks.
So he liked it. Did he put together the microscope?
No, we didn't. I told him we had to wait until you gave us instructions.
Yeah, there's some instructions on there, but it's kind of hard. So we're gonna. It's probably a little bit better to do that. So that's good.
He has like a little microscope, so he was using that. He was having, and then we did. I bought yeast at the beginning of COVID, like a whole ton of it. So we like activated that and smeared it on a dish, and then saw it grow, and then he put like different stuff on it, like lemon juice in one side and different kind of stuff.
Wow, you got a little scientist there. That's great.
Oh yeah, he's really into science right now. So, okay.
Cool. Anyone else? All right, so I'll be around. I'll be working. I'll leave my camera on. If I touch my nose or whatever like that, just ignore it.
Hey, Dr. Cap.
Yes, sir. Karamoko, how you doing?
All right. Hey, so I don't really have like a workstation or like kitchen space. So is it okay if I use like the other half of my desk?
Yeah, just so long as it's kind of like a flat area and stuff like that. And ideally, you want to be able to kind of sanitize it. But if you can, if you put down like some paper towels or something, work on that, and then throw out the paper towels after you're done, that would give you a good spot.
I just sanitized it pretty much with with this, and it's only like about that big half of my desk corner.
Yeah. No, that's more than enough. That's great.
Okay. I'm just trying to like work with what I got.
Yeah, and that's what we're trying to do. And actually, I like that you're doing that. So one of the things I'm trying to work on is we'll try and test a little bit how good our sanitizers are working.
You got it. And then I was going to ask you, do you want us like to put on our lab coats, goggles, and gloves and all that stuff, or is it really not necessary for this one? Okay, so I got it. All right, thanks. Now I got my goggles right here, but I was just like, if it's at home, do you want us to? Look, I got it. All right, thanks.
Yeah. So there's a difference between what you can do and what I have to say.
You got it. So I'm just gonna act like I'm in the lab and do everything accordingly. Thank you.
Yeah. Okay.
Excuse me, Dr. Pat.
Yes, ma'am.
So I'm just going back to my one of my previous human biology classes that I took when we did petri dishes, and I remember my group we used Q-tips to swab. We used my nails because I wear long nails. So to see how much bacteria is under there, so we use cotton swabs and then to rub.
If you have, so I didn't send you guys home with any cotton swabs, but if you have cotton swabs and you want to use them, by all means.
Okay, thank you.
Hey, Dr. Cap. Oh, there you go. Hey, Dr. Cap, can you hear me?
Yeah, I can hear you. It's a little noisy part of the lab. Let me go to the other part.
Okay.
All right, that looks good.
All right, so yeah, I'm set up now. I had to run to my car to grab my stuff. All right, so then I was going to ask you. So the first part says basically take a picture of our workstation. What I did was I sanitized everything down like on my desk, and then I laid paper towels over everything, and then I placed my stuff there. Do you want me just to take a picture of that, or do you not want the paper towels under the material?
Nah, just give me a picture of whatever you have. That's fine.
Okay,
Hey, can I get a picture of you?
sure.
Cool.
All right. So I'm just treating it like a regular lab. I even got my gloves set up on my stuff. So is that okay?
That's great. That's exactly what you want to do.
Okay, and then I'll take a picture of this. I even changed my clothes. No loose clothes, no anything. So I said, let me do this like I'm actually there with you. Okay.
Cool.
All right. And let me see if I had any more questions because I don't want to keep interrupting you. Let me see. Well, so if I'm working on this by myself, I remember you said we need 2 people. I don't have anybody else like here that can jump on this with me. I'm basically doing this alone. So that won't be a problem, right?
I'm sorry, what?
Oh, you didn't hear me? I said, I know that you said it'd be good if we had 2 people to, somebody else to work on this with us, but I don't have anybody else.
Yeah. No, that's fine. That's
okay.
one of the reasons why I'm here too. If you need some help, I'm here.
Okay. All right. Let me jump on this, and if I have a question, I'll jump back on. Thanks.
Okay, great.
All right, Dr. Cap. Another quick question. So I'm on number 2. It says you can open your Petri plate and let it stand to capture microbes as they move in the air, either falling into place or being pushed by a breeze. So I held my Petri dish in front of like my fan a little bit, and then I'm sitting it back on my desktop to see everything that it collects. But then it asked me to imprint a pin, a coin, or something onto the surface of the plate.
So you got 3 plates and you want to do 3 different things with those plates, right? So you can set one out and just let it sit. You want to let it sit for at least 5 minutes or so. And you're not going to see anything go on it now because they're microscopic. Can't see them with the naked eye.
Okay.
And so you're going to want to seal them up and let them grow for 3 days. But just take one plate out and put it underneath your fan. If you use a fan, just take one plate and put it out there. If you want to touch something, or if you want to put your pen on it or something like that, that's all good too.
Okay, great. So do you want me to do, so you want me to do one with a coin, one with a pen, and then one with the regular, right?
I want you to do whatever you want to do. You got 3 plates and you can do whatever you want as long as they're exposed to something in that area.
Okay, great. And then you said, and then after I do that, I should label them. I should actually label them before I do anything, and then let them sit for 3 days, right?
So you want to label them what they are, so you don't forget, right? So
No, I'm aware of the labeling.
plate number 1 underneath the fan for 5 minutes. Plate number 2 on the desktop open for 5 minutes. Plate number 3, tip of my nose.
You got it.
And then, so what you would do is, and then you would put them in a, put them in a container or something like that, and then kind of keep them in the dark. You want them to be at room temperature for the next 3 days. And then you'll pull them out and take a look at them.
Okay, you got it.
All right?
Yep.
All right, everyone in the class, it is about 9:27, right? So we have until about 10:30. We're going to start up lecture at 11 o'clock. I got a few things to do before then. I'll be kind of closing this down, but I will hang out. It's my office hour. If you want to talk to me, that's great. But any questions, comments, or concerns? Okay. All right, I'm going to stop recording.